Hello everyone, and welcome to Cell Culture Cafe, an interactive webinar series devoted to presenting answers and advice to common cell culture issues. I am your host, Megan Morlock, and today's presentation is Ultrapure Water and Cell Culture, Know Why, What, and How for Proper Water System Maintenance. Today's webinar presenter is Kim Knepper, the Global Application Specialist for Water Purification at Thermo Fisher Scientific. Kim previously served as technical product and sales support for the Barnstead Water Purification Solutions. Kim works with teams to develop training programs and perform application testing and analysis. Kim holds a Bachelor of Science degree with a major in biology. Before we begin, I would like to cover a few procedural items. This webinar is being recorded. As a registered guest, you will receive a link to this webinar in your email when it becomes available. This webinar, including Q&A, will last about an hour. Type your questions at any time into the text box in the Q&A module that you see on the right side of the screen. Kim will answer questions at the end of the webinar. This webinar is interactive, and you will be asked a few survey questions throughout the presentation. We will share the results so you can compare your answers with your peers. This ends our introduction, so without further delay, I present to you Kim Knepper.
Thank you, Megan. So in your water system in your lab is expected to deliver ultrapure water consistently. So this webinar is to help you understand your system and to be able to be a little proactive to avoid problems with the water system and any interruptions to your daily work. So in today's webinar, we're going to take that critical look at the ultrapure water system that is used in the cell culture lab. We will review the 6 impurities found in the lab water, including one that is critical to mammalian cell culture. Next, we'll discuss the common water system consumables and their critical role to purify ultrapure water. Understanding those different components in the water system will help identify why they need to be changed. We'll also point out guidelines for setting up standard maintenance for the ultrapure water system for consistent quality. And then lastly, as a bonus, we're going to talk about the types of water to use with general lab equipment that do not require ultrapure water, such as water for humidity in CO2 incubators and for water baths. But first, let's start with a survey question to find out more about your labs.
Thanks, Kim. Using the survey question module in the center of the screen, please answer the following question. In your lab, what applications require pure water? Select all that apply. Also, as a quick reminder, if you have any questions, you can enter those using the Q&A module in the lower right corner of your screen. And I'll give it another moment as there are answers still coming in. Okay, Kim, I'm going to broadcast the results and turn it back over to you.
Thanks, Megan. So I see a majority of you are making reagents and doing tissue culture, making media and buffers, and so that sounds great. And then there's quite a few of you that are filling water baths and incubators, so we'll be talking about water for those, and that will be helpful to you as well. And then a smaller percentage of you also do some analytical work in your lab. So why do labs need special water? So all labs need some water for the rinsing glassware, filling incubators and water baths, making reagents, and growing cultures. So why can't we just use tap water? Well, tap water has many impurities that could interfere with our daily experiments. And what about the central building water systems that are feeding the building or maybe a whole floor? Yes, even though they may be high impurity, they also have a lot of impurities that could affect critical work as well. So the more sensitive the applications in the lab, the more impurities that do need to be removed from the water. But even something as simple as filling a water bath may not seem as simple when there's a specific quality of water that's required by the manufacturer. When discussing water purification, it's important to review the different types of impurities in the water and the purification technology or the consumable they affect the most. So the first impurity we're going to talk about in today's webinar is the most common impurity in water, ionized solids. Now these are positive and negative ions from dissolved salts and minerals in the water, and they're easily measured by checking the resistance or the conductivity of the water. Now resistivity and conductivity are the same reading. They're just the inverse of each other. The higher the purity, the fewer the ions in the water. Now most of you may recognize the purity reading of 18.2 megohm resistivity, which is the theoretical highest purity of water. Now most commonly found ions are calcium and magnesium, which create water hardness, and bicarbonate ions that make up water alkalinity. Calcium, magnesium, and sodium all originate from the rock and the soil and the areas where the water passes through. Silica is another dissolved solid with a weak ionic charge, and that's commonly found in water, especially coming off of the regenerated service DI tanks. All of these ions can create a deposit on glassware or in equipment, and they can also interfere with sensitive buffers and trace analytical methods. Typical tap water ionized solids can range anywhere from 30 to 500 part per million. Ions are easily removed from the water by deionization, reverse osmosis, or distillation. Now, how can ions affect an ultrapure water system? Well, ions are attracted to the deionization resins. So the more ions found in the water, the quicker it will exhaust the cartridges. In reverse osmosis systems, hard water deposits from the calcium and magnesium can form on the RO membranes, shortening their life. And then in distillation systems, heating elements and interior components will have less efficiency and shorter overall lifetime. A second impurity in water are organic compounds. Total organic carbon, or TOC, come from sources such as agricultural runoff, decomposition of plants and animals, and industrial waste. And they can even come from plasticizers, the glues and dyes from plumbing materials. Now organics in the feed water can range anywhere from 1 to 5 part per million. In some cases, the TOC in the feed water can fluctuate during certain times of the year, especially for customers with a shallow well or maybe an open body of water like a reservoir. These impurities, organic impurities, can impact any of your experiments that will measure organics such as HPLC, GCMS, or TOC analyzers. Organics are removed by carbon or by UV lights, and it can be removed by reverse osmosis and by distillation. In the water purification system, elevated organics could plug carbon filters, they could foul RO membranes, and they could interfere with the deionization process. The third impurity in water are particulates. So particulates in water such as rust, sand, silt, plumbing debris, these actually may be visibly floating around in your water, or they may even settle out into your beaker. Now these particulates can plug valves and filters, and in reverse osmosis systems, it can plug the RO membranes. And even particulates that are submicron can have the same effect. Now particulates can be measured by turbidity or by silt density index, and they can be removed by consumables such as filters, even the process of distillation and reverse osmosis. For most water systems, the feed water turbidity should be below 1 NTU, or additional pre-filters may actually be necessary. The fourth impurity in water is bacteria, which is very common to find in water, and yes, even in chlorinated tap water and in ultrapure water systems. Bacteria and their byproducts can create problems with molecular biology applications, growing cells, and microbiology. Lab water systems provide a warm, happy home for bacteria to grow. And since it's easy for bacteria to contaminate a water system, there are multiple technologies used to remove bacteria from ultrapure water systems, such as filters, UV lights, and reverse osmosis membranes. Cleaning solutions are often used in the water system to reduce the bacteria levels as well. Bacteria can be measured in several ways, but the most common is by growing the bacteria on media and counting the colony-forming units. Now, bacteria in the system also creates two other impurities that we're going to talk about next. The fifth impurity in water are nucleases. The enzymes RNase and DNase can be a problem for molecular biology applications. Type 1 water systems that have an ultrafilter will deliver nuclease-free water without the hassle of the extra steps of using DEPC and then autoclaving the water. Water systems that combine ultrafiltration with the ultraviolet lights are an effective way to remove RNase, DNase, as well as bacteria from the water. And then the last impurity that is found in water that can interfere with the work of a cell culture lab are endotoxins, also known as pyrogens. These are the byproducts of gram-negative bacteria, and when injected into a human or an animal, it would induce a high fever. Pyrogen contamination can slow the growth of cells or tissues or interfere with other research applications. These are measurable in water with the bacterial endotoxin test, which is also called the luminous amoebocyte lysate test. Endotoxins are easily removed by the process of distillation, but they're also removed in ultrapure water by ultrafiltration, which is a very fine filtration down to less than 0.001 microns. Now, these ultrafilters can be found in the water pathway, filtering the water in the system as it's being recirculated, or it could be found at the point of use, which would be open to the atmosphere. So let's take the time to have another survey question to find out more about your water systems.
Thank you, Kim. Using the survey question module in the center of the screen, please answer the following question. What kind of water system is being used in your laboratory? If you have any questions, you can enter those using the Q&A module in the lower right corner of your screen. And I'll give it another moment as answers are still coming in. Okay, Kim, I'm going to broadcast the results and turn it back over to you.
Thanks, Megan. So I see that most of you have distillation in your lab, which is great. And then more commonly is also the type 1 system with a pre-treated water. And there's quite a few of you that are not sure. So this might be a good opportunity to find out what kind of water is feeding your lab. And then some of you are using a mixture of type 1 with tap water or type 2 and RO water. So let's review the different types of water purification technologies. And often you'll find that these need to be combined to purify the water to the highest standards to meet the critical needs of your lab. So distillation. By boiling the water to steam and then recondensing the water into another vessel, this allows a majority of the impurities to be removed from the water. So bacteria, endotoxins, most of the ions. So in this one technology, this can remove most contaminants very consistently. Now reverse osmosis, which is similar to distillation. So reverse osmosis can remove a majority of the impurities in the water, but it's a % removal technology. So therefore, it's important to note that the quality of the RO water is going to depend on the feed water and the age of the membrane. So maybe not quite so consistent as distillation. Now, although distillation and reverse osmosis can remove a large amount of impurities, they may be used as a pretreatment to a type 1 system. So deionization is used in the type 1 system, and these use resins to remove only the positive and negative ions from the water. Now, often deionization is not used alone in the ultrapure water system. It's often combined with other technologies to remove a wide array of impurities, such as UV oxidation, which is a great way to oxidize bacteria and organics, carbon absorption, which will remove organic compounds and chlorine from the water, ultrafiltration will remove nucleases, pyrogens, and bacteria, and filtration, a final filtration using an absolute filter to remove particles and bacteria. So next, let's talk about how some of these systems work. So as we mentioned, distillation in this one process produces water that is pure of ionized solids, bacteria, and endotoxins, and most gases and organics. Now, in a classic still, a boiler contains potable water that is heated to boiling, and the steam rises up through a baffle or a steam trap that forces the vapor through directional changes to further scrub the steam of any impurities. This effectively removes endotoxins and increases the resistivity. And this allows only the pure vapor to pass into the condenser where it is cooled and condensed back into a liquid state. The distillate is then collected in a storage tank or a storage vessel. Now, accessories could be added to this process to make it automatic and to filter and protect the water in the storage tank. Now, this process produces water slowly, and the system does require large amounts of water for cooling the steam, but is an excellent source of water for lab use. Next, let's look at the purification path of a type 1 system and how it works to purify water. So as the water enters the ultrapure water system, a great feature is the feed water conductivity cell to identify the quality of the water coming into the system. So if there is a breach in the feed water quality, the lab can be alerted, so either the feed water issue can be identified or the type 1 system can be shut off until the issue is resolved. Feed water problems can exhaust consumables very quickly. So next, we have the recirculation pump. And the pump recirculates the water through the system to ensure high quality of water on demand. The path for the polisher then sends the water past a UV light to oxidize organics and kill bacteria if the system has this feature. And the UV light is going to create carbon dioxide and release any ions attached to the organic compounds. From there, the water flows through a deionization cartridge with resins to remove ionic impurities, including those byproducts from the UV oxidation. Now, this is the most common consumable in the ultrapure water system, and its job is to achieve 18.2 megohm purity. So next, if the system has an ultrafilter for life science applications, the endotoxins, nucleases, and bacteria will also be removed. And at the dispenser, there's a 0.2 micron final filter. If it's not being dispensed, that water will recirculate all the way back to the very beginning of the system. Now, tap water as a feed water to a type 1 system often requires some sort of pretreatment. So it's common to use a reverse osmosis unit to do this. Now, RO is a great way to remove a broad spectrum of impurities. In a typical flow path for an RO system, you're going to notice some common components. So first, RO membranes require some type of pretreatment, maybe such as a pre-filter, a carbon cartridge, or some type of hardness reducer. From there, the water is then forced through the membrane by either the water pressure or an internal pump. Impurities are then collected on the surface of the membrane and then flushed to drain. The purified water will then continue on into the storage tank. The water could be used for general lab use, or it could be used as a feed water for type 1 systems. Now, note, RO systems do make water slowly, so a storage tank is going to be necessary to collect the water. And as mentioned earlier, RO systems are percent removal technology. Therefore, the water purity is going to be based or vary on the feed water or the age of the membrane. Now, customers with high ionized solids in their tap water are going to have higher ionized solids in the RO product water. So next, we're going to be introduced in more detail to the 7 consumables that we find in water systems and their critical role to purify water. And we're also going to review a typical guideline of when and how to change them. But actually, we're going to have a detour. We're going to start with distillation systems because they don't usually have consumables. However, hard water deposits will precipitate out and they will coat the inside of the boiling chamber and the heating element. So cleaning the still with acid will be necessary to remove that buildup, or over time it will shorten the element life or the boiler or the other components. Now, tubing going in and out of the still should be checked and replaced as necessary. And replacing the tubing, use the tubing recommended by the manufacturer, especially the product water. The water coming out of the still is going to be hot, over 80 degrees Celsius. Therefore, you need tubing that can withstand that high temperature without breaking down. So I've actually had cases where customers have used the wrong tubing that could not handle the hot temperature, and the tubing was scorching and breaking down, and they actually had a layer of oil on the top of their product water in the storage tank. The water in the storage reservoir should be turned over about every 2 days to prevent degradation and to keep the water fresh. Now, distillation systems may have some consumables as accessories. So, for instance, you may, instead of to reduce the frequency of cleaning, you may have a pre-treatment with some DI cartridges to remove the hardness ions before they go into the boiler. So note what kind of indicator is going to let you know when those cartridges need to be replaced. And then other consumables would include vent filters and UV lights for the storage tank. So just double check with the manufacturer to find out the recommended change-out schedule for those. But next, let's talk about the consumables that are typically found in a Type 1 point-of-use ultrapure water system. The most common consumable is the Ultrapure deionization cartridge. Deionization or DI cartridges are the backbone of the Type 1 system because they are creating that high purity, the 18.2 megohm display by removing ionized solids and gases from the water. And when these cartridges are exhausted, you may notice a change in the displayed purity. The purity may start dropping to lower numbers slowly at first and then dropping off quickly. So this is your signal that ions are getting into the product water and it will soon be time to replace the cartridge. Now, some systems allow you to set an alert on the display if the purity has dropped below your acceptable levels. The range of acceptable purity is really going to depend on the sensitivity of your methods and your applications. A common question we get is, how long will the deionization cartridges last in my water system? And this is a great question, but it really depends on 2 critical factors: the quality of the feed water and the amount of water processed through it. Therefore, if the feed water source is good and well maintained, the cartridge life could be longer. And also realize that every drop of water used by the water system will have used up some of the DI cartridge life. So if you're flushing large amounts of water through the system before you take your sample, that could impact some of the life. Now, sometimes with low usage and a really high quality feed water, cartridges could definitely last longer than a year. So it's a good practice to change the DI cartridges and disinfect the system at least once a year. Dual wavelength UV lights in an ultrapure water system do 2 things. They destroy bacteria and they break down organic compounds. So if the intensity of the UV bulb is reduced or the bulb is burned out, it will not be effective at keeping bacteria and total organic carbon to the lowest levels. The UV lights are usually replaced based on time, with the system giving a visual or maybe an audible alert. Now, some systems will display an error message if the bulb falls below a certain intensity or if it's burned out. Now, thermoscientific engineers designed a photoelectrode system that measures the UV light intensity to verify when the UV bulb should be replaced in units that have a TOC readout. So always check with the manufacturer instructions to be aware of how your particular system signals you to change that UV bulb. Now, when changing the UV bulb, there's a few things to remember. You follow the step-by-step instructions. But when handling the bulb, it's a good practice to wear gloves, as oil and dirt from your hands could affect the intensity of the bulb. And sometimes you might see the glow of the UV bulb in the tubing or in the fittings, and it's nice and blue, but never look directly at a UV light if it's on. Be sure to have UV protective lenses or glasses. And UV bulbs are protected in their housings with a quartz sleeve to prevent the direct contact with water, so make sure that sleeve is intact and sealed. The third consumable in ultrapure water systems are ultrafilters. Now, ultrafilters are very fine filters that are designed to remove pyrogens or endotoxins, nucleases, and bacteria. When they're built into the water system, they can last up to 2 years. These ultrafilters have a large surface area and usually a reject or a rinse line to flush the surface of the ultrafilter of any buildup of impurities. So you would change the ultrafilter when you have an endotoxin breakthrough that doesn't go away after disinfection, or if there may be a reduced flow rate. Now, tips on changing the ultrafilter would include locating it in the system, because in some cases it may actually be in the back of the unit. And since ultrafilters may be replaced about every 2 years, you might need to refer to the detailed instructions for proper changing. Inline ultrafilters have 3 connection points, so make sure to mark the tubing if necessary to ensure that the new filter is installed correctly. And then disinfection of the system is often recommended, and this will also clean the ultrafilter as well. So if the system is down for any period of time, some ultrapure water systems also have a manual rinse function to help rinse that surface of the ultrafilter for any buildup. Now, ultrafilters do have a lot of surface area, so new filters may need to be flushed to purge any trapped air and to wet the surfaces. The final filters are the last contact the water has with the ultrapure water system. The 0.2 micron filters are important to pick up any of those last particles, bacteria, or resin beads from new cartridges. The water is not recirculated through the final filter because these are at that point of use of dispensing. So therefore, frequent changes are going to be important. And it's recommended to avoid attaching any additional tubing to the final filter, as it could be a source of contamination and growth. But if you must add tubing for more convenient dispensing, keep that length to a minimum, use clean, inert materials, and replace it often. Change the final filter when you see that reduced flow rate, indicating that the filter is getting plugged, or if you would see bacteria breakthrough. And then always change the final filter when you change the DI cartridge or if you disinfect the system. Now, as we mentioned earlier, if the feed water is tap water, often some type of pre-treatment of the water is going to be used to remove a large amount of impurities so that ultrapure water system doesn't have to work so hard. So since RO systems are very common to use as a pre-treatment, or maybe even as a purified water source by itself, then we're going to next look at the consumables that are found in the typical RO system. Now, RO membranes remove up to 98%, 99% of the impurities in the water. But as the RO membrane ages or fouls, the flow of the water through the membrane can change as well. and so will the percent rejection of the salts. As more impurities come through the RO product water, this is going to affect the quality of the water in the storage tank, and it could also affect the Ultrapure water system cartridge life. So if the type 1 system in your lab is going through cartridges quickly, it is a very good indicator to check the pre-treatment or the RO system performance. Now, some RO systems do have a digital display or a light indicator for the percent rejection and alerts the users of the product quality changing or dropping below a certain level. But what else should be monitored in the RO system? Well, double check with the manufacturer for instructions for the system's capabilities, but common things to check on a regular basis are feed water quality, temperature, flow rate, and product water purity. Now, RO membranes tend to change over time, so by monitoring the system, you're going to be able to respond to these changes before that purity reaches a critical level. Now, RO membranes are typically replaced about every 1 to 2 years. And again, it's important to check the RO system on a regular basis to verify that the RO membrane is performing to specification. Now, to change the RO membrane, you would follow the recommended steps in the manual, or you could schedule a service visit. And it's important, if you're going to be changing it, important to note the orientation of the membrane in the system before removing the old and installing the new. RO membranes do have a preservative that must be flushed to drain before use. So this flushing of the membrane could take anywhere from 2 to 4 hours. If any water is collected in the tank during this time, it really should be discarded. And then once the system's been flushed, you can check the product and the reject flow rates and make any adjustments to the system as necessary. So RO systems, as we said, do require some pre-treatment to protect it from the typical tap water contaminants. Now, RO pre-treatment can be built into the system, or it could be found externally in the water line before the system. And different cartridges or filters are utilized to deal with the different problems that are in the particular water supply. So how important are RO pre-filters? Well, they're very important. The RO membrane is doing a huge job by removing over 90% of the impurities in the water. So we want it to do its job effectively for a long time. So what are those 3 impurities that could damage the RO membranes? The first one is particulates. Now these can tear holes in the membranes or block the fine pores. And these are easily removed by particulate filters. Now these filters are replaced when the flow or the pressure out of the pre-filter is reduced. If the system has pressure gauges for the pre-filters, note the pressure changes over time to know when the filters will need to be replaced. Now, a good practice would be to replace them on a regular basis before that flow or the pressure is compromised. So next is the water hardness. Now, hardness formers in the feed water, like calcium and magnesium, will damage RO membranes. Water softeners or hardness stabilizers are used to reduce the effects of the hard water. Now, water softeners will ion exchange the calcium and magnesium in the water for softer sodium ions, and then periodically backflush the softener resins with the salt water to regenerate them. So this will require replacement salt pellets in the brine tank on a regular basis. Now, hardness stabilizers, they actually bind with the calcium and magnesium in the water and prevent them from precipitating out of the water and onto the membrane. So these are crystals, and these cartridges, the crystals in the cartridges will dissolve slowly, releasing the stabilizer into the water. So it's best to change the hardness stabilizer cartridges when the crystals are about half gone. And then lastly, we have chlorine. Now, while there are some RO membranes that do require chlorine to prevent bacteria from damaging the membrane, most RO membranes cannot tolerate chlorine. So double check with the manufacturer for the kind of membrane that is used in your system and what's the suggested pretreatment. For membranes that cannot tolerate chlorine, it's often removed with a carbon prefilter or pitting of the membrane could occur. Now, carbon cartridges would need to be replaced on a regular basis based on a time or an alert on the water system. But also note, there's in some labs that have a water supply on a surface source. You may have experienced elevated organics during certain times of the year, like the spring or the fall, so that may affect your carbon cartridges. So the last consumable that could be found in a water system are vent filters, or accessories that are associated with the storage tank. Now, vent filters or CO2 vent filters are used to protect the stored purified water from airborne contaminants like bacteria, dust, or even CO2. Now, some storage tanks may even use a UV light to discourage bacteria growth. So refer to the manufacturer timeline and instructions for replacement. The stored water in a tank should be turned over on a regular basis to discourage bacterial growth and to keep that water fresh. Other maintenance tips would include checking the tubing going in and out of the tank to ensure that none of the tubing has any obvious growth or it's not getting weak. And ensure that if there's an overflow line on the storage tank, make sure it's able to flow freely to a drain that's open to the atmosphere and that it's functioning properly. So let's take a second to look at another survey question.
Thank you, Kim. Using the survey question module in the center of your screen, please answer the following question. Do you have a standard operating procedure to change consumables for your water system? Again, as a quick reminder, if you have any questions, you can enter those using the Q&A module in the lower right corner of your screen. And I'll give it another moment as answers are coming in. Okay, Kim, I'm going to turn it back over to you.
Okay, thanks, Megan. So this is interesting. There's a split between that, yes, some of you have replaced the consumables on a regular basis, and then half also don't have any kind of replacement plan that you replace them when you notice a problem or if there's an alert on the system. And then a small percentage of you that are not sure. So it'll be good to talk about the water system maintenance, and hopefully after today's webinar, this will give you a good plan for maintaining the system. So what are some of the common tips used by labs that effectively maintain their water systems? 1, create a list of consumables that are used in the water system, including the description and the ordering number. And then also identify the consumables that are used in any accessories and include anything necessary for cleaning the system. And then also keep a log near the water system. So this would include a list of what consumables to replace, when they were replaced, and who performed that maintenance. 2, understand and recognize when to change the consumables. So is there an audible alert? Is there a visual signal? Or is it just simply based on time? And then 3, monitor the system's purity for any changes and alerts. So some water systems are designed to help you know when to take action before the purity drops to critical levels. But sometimes changes happen gradually over time, so it's good to be aware of the system's overall performance. Now, all water system manufacturers establish guidelines for the consumable's life, but let's face it, things happen. Water is fluid, dynamic, and it can carry all kinds of impurities that could exhaust consumables unexpectedly. So be prepared and have consumables on hand just to make sure that you can react to those occurrences. So what would be a typical change-out schedule? So in some cases, it would depend on the feed water and the usage of the system. But a good general rule of thumb for pre-filters for tap water, they're going to need to be replaced about every 4 months for average use. But if the lab has heavier usages of water, or if there's seasonal changes, they may need to replace those more frequently. Don't skip this step. The RO pretreatment is critical to the health of the entire rest of the system. Now, RO membranes, they can last up to 2 years. However, depending on the feed water quality, it could be shorter, it could even be longer. UV lights and ultrafilters could last up to 2 years. Tank filters could be replaced yearly. Ultrapure DI cartridges need to be replaced a minimum of once a year or based on the purity display. And then final filters need to be replaced when the flow of the water is reduced, or change it when the DI cartridges are replaced, or when the system is disinfected. So why should disinfection be done on an ultrapure water system? Yes, bacteria can live in an ultrapure water system. It is a warm environment with DI cartridges with carbon that provides food for the bacteria if the system's not maintained. Now, water systems with recirculation, that does help reduce the bacteria growth, but it does not remove all the biofilm. Systems with the UV light will also help oxidize the bacteria in the water in the flow path, but disinfection still needs to be done to help disinfect the entire system. So what should be done as just an annual maintenance? So it's good to disinfect the water system at least once a year. And this is especially good to do when you change the DI cartridges as well. Again, just to keep that biocontaminants in check and to flush the system completely. Typical steps in this process would be to remove the DI cartridge and the final filter and dispose of those. Install the water system cleaning cartridge in solution. You'll enter the cleaning function via the control panel, and then allow the system to do the disinfection process, which would include recirculation and flushes. Now, this cleaning should apply to any kind of storage tanks that are feeding the ultrapure water system as well. Again, just follow the operation instructions from the manufacturer for that complete procedure. Now, after cleaning the system, you're going to be ready to install a new cartridge and final filter. We do not recommend reinstalling the old DI cartridge because it possibly could have bacteria that would recolonize in your water system again. When you're handling the cartridges, have clean hands or maybe wear gloves, especially handling cartridge components that are going to be directly in contact with water. Now, some systems may require a timer, resetting a timer, or maybe entering a serial number of the cartridge to confirm that a change has been made. And if you're cleaning the storage tank, make sure to change those vent filters to ensure that protection of your stored water. And don't forget to, again, check and replace the RO system pre-filters in the water line as well. Okay. So we're going to be moving into our next section of the webinar. So let's start with a survey question.
Great. Thank you, Kim. Using the survey question module in the center of the screen, please answer the following question. In your lab, what is the source of water used for the general lab equipment, such as CO2 incubators and water baths? Again, as a quick reminder, if you have any questions, you can enter those using the Q&A module in the lower right corner of your screen. And again, I'll give it a couple of moments as answers are still coming in. Okay, Kim, I'm going to broadcast the results and turn it back over to you.
Thanks, Megan. So I see that about 50% of you are using water from essential supply, such as a DI, distilled, or RO supply, and then 38% of you are using a distilled water, and then a small percent are actually using type 1 water. So it would be good, we'll be talking about all these different types of water. So what if my incubator or water bath is made of stainless steel. Isn't it corrosion resistant, therefore should not corrode or rust? Actually, no. Stainless steel is corrosion resistant, not corrosion proof. And I'm still seeing the results up on the poll questions, Megan. Thanks. So stainless steel is corrosion resistant, not corrosion proof. So it is susceptible to corrosion like anything else if it's not treated properly. It has pits and pockets where water could sit and corrode. It is susceptible to certain chemicals or even scratches and rough handling, even sliding incubator shelves on integrated shelf supports, anything that could ruin that protective oxidative layer, which could be common to stainless steel. And then certain different types of water possibly could cause rusting and corrosion. So what kind of water can I use in my general lab equipment, such as incubators and water baths? Well, tap water just has too many salts and minerals, and that can precipitate out of water and create deposits and with the evaporation of the water, and that could cause rusting and corroding. Now, Ultrapure type 1 water, while it's free of salts and minerals, is too pure. The lack of ions make Ultrapure water aggressive, and it tries to absorb anything it can back into it, including any ions it can find from the container that it's held in and CO2 from the air. Now, CO2 will lower the pH of the water, making it hard on the stainless steel as well. So what is just right? Distilled water. Distilled water is often the most often recommended water as to be used for the general lab equipment. So what is it about distilled water that makes it so preferred for feeding an incubator or water bath? What is so special about it? So distilled water is great because it is naturally sterile and it does not have impurities in it to create deposits or problems with the stainless steel. What makes distilled water optimal is because of the process of distillation is slow. So while the water is being collected into a storage container, the distilled water has time to reach an equilibrium with the atmosphere and with the container, making it less aggressive. So while the distillation system may be producing well above 1 megohm purity, the water in the storage tank could easily be 0.5 to 1 megohm in the storage tank. You can make distilled water fresh in your lab, or you could purchase bottled water that's been sitting on a shelf. But note, distilled water is not the only option for using in the general lab equipment. So deionized water. So everyone gets a little scared when we talk about deionized water and stainless steel and lab equipment. And the fear is because we get so used to assuming deionized water is type 1 water and that it's highly aggressive. The water at that purity is not its natural state, and again, it wants to absorb ions back into it to reach that equilibrium. But when we talk about deionized water, it is a method of purification. And not all DI water is ultrapure type 1. Now, some DI water is actually the same quality as distilled. So there's different levels of purity that can be achieved by deionization. And the lower the purity, the more ions in the water, and therefore less aggressive it will be on the lab equipment. So ASTM type 2, type 3, and type 4 water can be made by deionization, distillation, reverse osmosis, or even a combination of those purification methods. Now, note, while manufacturers may have a very specific resistivity or conductivity range that the water must be in to be used with the lab equipment, like for example, Thermo Scientific incubators have a recommendation of the water to be 50,000 ohms to 1 megohm quality. So and it's often kind of difficult to zoom right into that exact purity with even a simple DI system. But with brand new cartridges, the water, even a simple DI system could be above 1 megohm. But also note, in some cases, because the lab equipment will tell you if your DI water is too pure. So in some water baths or incubators, the water level sensor that tells you when to fill the water pan is actually a conductivity sensor. So the ions in the water will make a connection between the electrodes, and when the water gets too low, there's no connection and the alarm goes off. So if you're using ultrapure water or type 1 water in your incubator, the water sensor alarm could go off, indicating, hey, your water's too pure. So what can I do if all I have is deionized water in the lab? So if the DI water in the lab is too pure to be used in your general lab equipment, there are some steps that can be taken. So one option would be to add a neutral salt to increase the ionic strength of the water. And it really won't take much salt to add ions into the water. And it is recommended that if you do this, to replace the water and wipe out the unit on a regular basis, just so you don't get any buildup of salts due to evaporation. But another better option would be to add a sterile solution of weak sodium bicarbonate to add ions in the water to decrease that aggressive nature of the pure water. And it will also bring the pH between 8 and 9, which will tend to be non-corrosive. So for example, a 0.001 molar solution of bicarbonate will give you a pH of 8.27. So that can be achieved by adding 84 milligrams of sodium bicarb for every liter of DI water in the incubator. Now this would be expected to give you a solution conductivity of like 100 microsiemens or 0.01 megohm, which would be like a clean surface water. And again, change that water in the incubator and water bath, clean it, change it, and clean it on a regular basis. Now, is RO water quality high-quality water? Is RO water high-quality water and therefore aggressive like DI water? Again, since RO water is a percent rejection technology, so that purity is going to depend on the feed water and the age of the membrane. So the RO water may be acceptable water supply depending on the purity. But usually RO water in itself is not ultra pure, and in some cases it may not even be pure enough. So in summary, tap water or central pretreated water systems may contain impurities that could interfere with growing tissues and cells in critical lab applications or causing deposits on instruments or glassware. The water systems use several technologies, including filters, membranes, cartridges, and UV lights to remove the impurities in the water. And the purity of the water does depend on the proper maintenance of the water system. So while establishing standard maintenance of the water system, it's important to know what consumables are in the system and to be aware when they need to be replaced. And then changing the consumables in this water system on a regular basis helps ensure that high-quality water when you need it. And then lastly, when using water to fill general lab equipment, such as water baths and incubators, double-check with the manufacturer on the recommended quality. And usually distilled water is highly recommended, but if the available water supply is too pure, then we can take some steps to lower that purity so it's not corrosive to your equipment. So as we come to the question and answer portion of this presentation, I'd like to point out to please be sure to visit our resource center at thermofisher.com/purewater to view our line of water purification systems and to access our product videos, brochures, application notes, and presentations.
Thank you, Kim. And it looks like we've received several questions, and please keep them coming. But before we get to questions, I'd like to draw everyone's attention to some links now on your screen. The first is an attendee survey found in the lower left corner. Let us know what you thought about today's session and suggest future webinar topics. Also, at the bottom of the screen, we've included several resources related to today's discussion. We encourage you to download those for future reference. Okay, let's dive into some questions, Kim. All right, the first question is, can the bacteria get out of the filters?
Can the bacteria, so bacteria can get out of the water system through the final filters. They could eventually grow out of the final filter if they've been there a while. So that is definitely possible, and that's why it's recommended to replace that final filter on a regular basis.
Okay, great. Your next question is, how should I dispose of the used filters?
So often these could be disposed of in your normal disposal, your garbage garbage that you for your lab. In some cases, some cartridges may be recyclable. The plastic might be recyclable, but typically they're completely disposable.
Okay, your next question is, how do I know the purity of my RO water?
So either the system will measure and have a display to tell you the purity. In some cases it may not. So you could get a conductivity meter to test the quality or measure the quality of the water as it's leaving the system, going into the storage tank.
Okay, Kim, we have a couple more for you. We have a really good building water supply. The Type 1 system cartridges have not been replaced in a couple of years, and the system still reads 18.2. Should we change our new cartridges?
So good question. So yes, while sometimes cartridges can last way over a year, the display of the system may only be reading the resistivity at 18.2, but it's not measuring other parameters in the water that may be elevated. So you may have higher levels of organics or even bacteria or pyrogens, and it's not giving any kind of measurement of that. So it would be a good idea, just as standard maintenance, to start fresh with a clean system once a year and to change those cartridges.
Okay, next question. Does it make sense to disinfect the Type 1 system without changing the DI cartridge? Will that help reduce the bacteria in the unit?
So while disinfecting the system will definitely clean the system and help kill any bacteria that would be in the system, by the cartridge that's been in the system does have resins, DI resins and carbon that could be food for bacteria, and there may be bacteria growing in the cartridges. So if you disinfect the system and install the old cartridge back in that potentially could have bacteria, you could easily reinfect the system. So it really is a good choice to start fresh every year.
Okay, great. It says, our water system display reads around 1 or a little higher than 1. What type of water is this?
So that would be could be considered a type 2 or type 3 water supply. So yeah, so it would kind of fall between the type 1 and type 3. I'm sorry, type 2 or type 3.
Okay, Kim, one final question for you. How do you deal with bacteria smaller than 0.2 UM?
So for bacteria that would be smaller than 0.2 micron, that's where the ultrafilter would help remove those kind of bacteria, and it would trap them at that surface.
Okay, great. A big thank you to Kim and to all of our attendees for joining us for today's webinar. Please feel free to send any further questions to the email address shown on your screen. And if we did not get to your question, we will directly reach out to you. As a reminder, as a registered guest, you will receive an email within the next few days with a link to the recorded webinar. We encourage you to visit thermofisher.com Cell Culture Cafe for future webinar details and to access our library of recorded and on-demand sessions. Thanks again and have a great day.